Hello, my name is Peter Nivert. I'm a senior biomajor working in the Department of Biological Sciences under Dr. Papalsikis. I will be presenting my poster characterization of mammalian extracellular vesicles. One of the main focuses of our lab is the characterization of extracellular vesicles, which are directly related to cell-to-cell communication via the transfer of RNA, DNA, and protein cargo. They come in two major groups, exosomes, which are formed from the endosomal biogenesis, as well as microparticles, which are formed from the budding of the cell membrane. The characterization of them involve understanding which proteins were present in or on the membranes. We did this by isolating exosomes via centrifugation as well as ultracentrifugation and introducing magnetic beads, which would have antibodies attached to them, which would hopefully associate with four proteins that we're anticipating being present: CD81, CD63, annexin A1, and TSG101. We we had several controls. We had just beads. We had an IG control to make sure that the antibodies we introduced to the exosomes weren't just binding non-specifically. And we also had a secondary antibody only sample, which was just the secondary antibodies that we would introduce without the exosomes to make sure they were actually binding to the primary antibody like we were anticipating. The secondary antibody has a fluorescent tag on it, so we were able to detect it on flow. The results showed that there was a clear shift in the fluorescence intensity for CD81 as well as annexin A1, which indicated they were in high abundance on the exosomes. CD63 and TSG did not produce a major shift, which means that they are likely not present. Microparticles are extracellular vesicles are important because if we can load whatever cargo we would like into them, that would give us a quick delivery service to cells so that we can introduce whatever we'd like. Um, Another focus of our lab was the generation of a stable cell line, which can produce GFP and Cas9. Uh, Essentially, what we did was we endogenously loaded two different plasmids, one with an NLS tag and one with out the NLS tag. The NLS tag is just the nuclear localization sequence, which would mean that once the protein was created, it would get shuffled to the nucleus. One of our end goals was to hopefully have this protein complex enter microparticles so that we could deliver to cells and hopefully edit those genes. So the NLS tag was not anticipated to help very much. It would likely wind up just in the nucleus and not make it out to the microparticles. Our results showed that over a 4-day period, the NLS free tag um, had high expressing of the protein throughout all 4 days, as well as um, a clear indication that they were entering the microparticles. The NLS tags also had high expressing, but not as long as the non-NLS tags. And it did also wind up having protein in the microparticles, which means that the NLS tag does not exclusively keep it out of the microparticles. It doesn't get as high as without the tag, but it is nice to know that the NLS tag can enter microparticles. But that's all I have. Thank you for coming and listening to my poster.